So good afternoon, everyone. I'd like to welcome you to the Office of the Chief Scientist Science Seminar Series. I'm Andrew Paul. I'm a Senior Science Advisor with the Office of the Chief Scientist, and I will be your host for this winter's series of seminars. On the behalf of our entire team, I'd like to welcome you to our first seminar of the year. The purpose of these seminars is to exchange knowledge on interesting and relevant science that's happening across Alberta using an open learning and sharing environment. Today we have people joining us from across Alberta, and I want to acknowledge that we are on the lands and gathering places for Treaties 4, 6, 7, 8, and 10, as well as the homeland of the Métis. This includes the traditional territory of the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, and Saulteaux nations. So wherever you find yourself today, I encourage you to recognize, acknowledge the relationships with First Nations, Inuit, and Métis, and the land upon which we live, work, and learn. So today, to maintain our call quality, you'll see that your video and audio is being disabled. You can enter questions or comments that you have during the presentation at any time. But we'll save those comments and questions until the presentation is concluded, and I'll read out those questions, comments to our presenters in the order that they were received for them to respond to. And then finally, the last thing I just want to note for today, and that is we will be recording the seminar. These recordings are available on the Alberta Environment and Protected Areas YouTube page. So if you have colleagues that weren't able to attend today, please encourage them to go to that YouTube page, and they'll be able to pick up the recording of today's seminars. So with that, I would like to introduce our speakers today. We're welcoming Dr. James Wasmuth and Dr. Viraj Muthye from the University of Calgary, and they're going to be presenting on challenges posed by whirling disease in Alberta. Dr. Wasmuth uses genomics and computational biology to understand how parasitic species successfully infect and alter their hosts. He completed his PhD from the University of Edinburgh, moved to Toronto in 2006, and then took a position in the Faculty of Veterinary Medicine at the University of Calgary in 2011. His lab at the university has projects on blood-sucking worms in cattle, zombified ants, and parasitic jellyfish, which I'm sure they must have great Halloween parties. Dr. Muthye is a bioinformatician who has been exploring the evolution of invertebrate animals like jellyfish and sponges using computational techniques. He completed his master's in biotechnology from Northwestern University in Illinois and his PhD in bioinformatics and computational biology at Iowa State University. Viraj is now at the University of Calgary and is sequencing the genomes of 2 parasitic jellyfish that pose a significant threat to fish populations. And that is what they will be talking about today. And with that, I'm going to turn it over to Viraj and James. Thank you.
Thank you very much. And thank you very much for the invitation to speak today. Thank you for so many people coming along. I just want to say I'm currently actually on sabbatical and living in the south of France. So it's 9:30 my time, which is actually perfect for me because the children are asleep. So hopefully they won't just wander in. But thank you everyone for attending. Viraj will be doing most of the speaking. I'm going to give a quick introduction and about parasitology research or parasitology programs here in Alberta, as well as a bit about my own lab, and then Viraj will be talking more about the parasitic jellyfish project. He also controls the slide, so I'm going to ask him to go to the next slide for me, please, Viraj. So Viraj and I are both members of the Host Parasite Interactions Research Training Network here in Western Canada. I just want to say a few things about that because I'm sure many people don't know we exist, but we're a very enthusiastic community established in 2012 through an NSERC Create grant. Those are the $1.6 million grants to kind of create a new training program that focuses on professional development of students and postdocs, research collaboration, and we've done a lot with engagement in the public. So the HPI training program now features 20 labs, most of them at university, one at the AFC down in Lethbridge, across University of Victoria, U of A, U of C, University of Lethbridge, and out at the University of Manitoba. If you're interested in what we get up to, there's our web address, ucalgary.ca/HPI, and we're also on social media. Viraj, can you go to the next slide, please? So as well as professional development and research collaborations, one of the things that our students and postdocs have really excelled in is communicating the wonders of parasitology to the wider public. So I've got 2 slides with some photos about our activities. I think it's in your top left is the big group photo. That's the boot camp. So that's the professional development. That's some of the people in the program. We were out at the Kananaskis field station, and we go there for 2 days to help, build the networks of our students. We do training in writing, communication, and team building. And then, so if we go clockwise on the right, this are some of our students going to a local school where they talked about parasites and plants. Then on the bottom right, that's very small in the bottom is Chen-Hua Li, a former PhD student of mine. She's at the Contemporary Calgary Art Gallery, talking to about 150 members of the general public about the wonders of zombified ants. In the middle is me at the Science Center during one of their adult-only nights, where we filled it for about 400 people talking about parasitology. On the left is a recent event which includes Viraj, where they were at the Bow River Habitat Field Station talking about fish parasites. Next slide, please, Viraj. And then here's just an example of some of the materials where we try and fascinate the public. So on the left, fantasy animals get parasites too. This was done at the University of Alberta, and in the middle, those are paper mache ticks. So on the right is the before, on the left is the after, and in the middle is a horse ascarid worm. So we take those around schools and, freak out the children. Actually, we freak out the parents. The children love it. And then on the right, that is a crocheted tapeworm. So it's very, very big. And we use that to show how amazing the worms are and all the mouth parts and everything. We also run a blog. If you want to hear more about parasitology, then you can go to the parasiteblog.wordpress.com. A recent post, and this is our trainees are writing this, fascination, disgust, and social media, how parasitologists can fight against medical misinformation online. So our students and postdocs really take their learning and their experience in parasitology and try and apply this to the widest possible audience. Next slide, please, Viraj. So that's the HPI program. A little bit about my work. So I'm at the University of Calgary in the Faculty of Veterinary Medicine. This is a picture of some of the people that are in the lab. In purple is Viraj. At the bottom was our Christmas party when we went axe throwing. I want to say thank you to the people that pay the bills. So NSERC, Alberta Ministry of Environment and Protected Areas, ARDA, which is the Alberta Agricultural Funding Agency. Alberta Innovates has helped support the work, and we've had funding through the University of Calgary. Next slide, please, Viraj. So research in my lab, 2 of the big themes in the lab are genome to drug. So we do a lot of genome sequencing. If you don't know what that is, don't worry, Viraj will take you through it. But we try and understand how parasites evolve and how they interact with their host. And can we potentially use this information to treat the parasitic infection? So there's the genome to drug, where we go from the genome and we use a lot of computational techniques to try and come up with potential drugs. And then more from a kind of basic science perspective, host-parasite interactions, host behavior modification, which I'll touch on very briefly in a second, as well as molecular mimicry. So how does the parasite evolve proteins that look like ours that then messes with our immune system? So Viraj, can you jump to the next slide, please? So this was a question I was going to ask for people to answer in the chat, but I'm not going to because I realized that my biog says that we work on all three of these, so that probably answers it. But anyway, the question is, which of the following are found in Alberta? Blood-sucking worms, zombie ants, and parasitic jellyfish. The answer, as Viraj on the next slide will show you, is all three of them. So I'm just going to quickly go through the first two. So first of all, blood-sucking worms. Viraj, next slide, please. So if you're driving through Alberta, in addition to the cows, you may see some sheep. You'll see lots and lots and lots of sheep, and inside them are a bazillion worms. So on the right, hopefully on the right, but anyway, the thing that looks like a worm is Homunculus contortus, the barber pole worm. It's very small. It lives in the intestine of sheep. And the reason it's red is because it punches a hole in the intestine of the sheep and drinks the blood. So next slide, Viraj. The reason it's called the barber pole worm is because you can see wrapped round kind of the darker red piece in the worm. That's actually its intestine filled with blood. And the idea is that it looks like the pole that swirls outside the barber shop. Next slide. What you'll see here is, so if any of you have done a biology degree, you'll know about Caenorhabditis elegans, the genetic and developmental biology model. Natural habitat is either a petri dish in the lab or in rotting fruit in an orchard. So that's on the left-hand side, and you'll see its mouth parts at the bottom, which are very good for sucking up bacteria. On the right is Homunculus contortus, and you'll see its mouth parts there, where it's got that lancet, which jabs into the intestinal wall of sheep, and it's got that straw, so it's almost like a straw in bubble tea, and it sucks the blood out of the sheep host there. So we study not necessarily the morphological differences, but the genomic differences. How do you go from a free living nematode like C. elegans to a parasitic one like Homunculus contortus? Next slide, please, Viraj. Moving on to zombie ants. Next slide. So many of you will know about Cypress Hills Interprovincial Park, and I hope that many of you have gone. Cypress Hills Interprovincial Park is down in the southeast corner of Alberta and spans Alberta and Saskatchewan. And if you go down there from late spring to early fall, you might see this. Viraj, next slide. You might see, so these are pictures taken down there, ants clinging onto flowers and petals and bits of grass, and those ants are clamped there and do not want to let go. If you try and pull them off, they are holding on so tight that you actually may detach the body from the head that's still clinging on there. And the reason it's doing that is it's infected by a parasitic worm. Next slide, Viraj. Called Dicrocedium dendriticum, the lancet liver fluke. The reason it's called the liver fluke is because of its life cycle. So Viraj, next slide. So what we'll see here is its life cycle. So we have to start somewhere. So we'll start off with, so one of the hosts is cow, deer. It gets pooped out. A snail comes along, digests the poop, which is infected with the parasite. The snail becomes infected. Eventually it coughs up a big slime ball. The ant then goes and finds the slime ball, eats the slime ball, and then when the temperature drops in the evening, the ant, instead of going back to its nest, will climb up a blade of grass or onto a petal and wait there to get consumed by a passing mammal. So that's how it does the life cycle. The thing we're interested in, next slide please, Viraj, is one of the projects in the lab is understanding how the parasite mucks about with the behavior of the ant. So on this is a slice through an ant that's infected. The head's on the left, the gaster's on the right. So what you'll see in the gaster, or the butt part of the worm, is you'll see all these cysts, and those are most of the parasites that sat there. But one of them is where that red dot is towards the head, and that's the one that's controlling the the behavior change in the ant. And we've been doing some work with Cam Goat down at the University of Lethbridge in trying to understand what's the molecular signals that control that behavior change. There'll be some papers coming out soon, hopefully, on that. Next slide, please, Viraj. Just want to say that parasitic worms are what we mostly work on. Viraj will be talking about jellyfish. They're everywhere. You might recognize this as the World Health Organization logo. That's not a snake. It's really a worm. Next slide. It's the guinea worm. So what that is supposed to, and there's differences of opinion, but the correct answer is that that is actually like the stick where you're pulling the guinea worm out of someone's leg. Next slide, please, Viraj. You'll see that emblem most places. So as I said, I currently live in the south of France. Recently, we were in Carcassonne, and by a pharmacy. On the right, you might see something that looks like the WHO logo. I see a worm that's by a cocktail glass. And I've seen this kind of badge else places in France, which might tell you something. And then on the left is a photo I took while we were in New Zealand. And this was in a toilet in a cafe in the center of New Zealand. I got worms. I'll tell you how I got them. I didn't wash my hands, and now they're in my bottom. Excuse the grammatical errors in there, but that's just to say that parasitic worms are everywhere. They're in cattle. They're in they're in the sheep. They're fortunately they're not in us anymore, and they're also in our public imagination. Next slide, please, Viraj. So I'm now going to hand over to Viraj because we also work on parasitic jellyfish, and that's where Viraj comes in. Viraj.
Right. So, hi everyone. My name is Viraj, and I'm a postdoc in James's lab. And today I will talk to you about some of the really interesting stuff, research which we are doing on Alberta's parasitic jellyfish. So I do want to start this presentation by giving my acknowledgements first to James for his constant support, and then a lot of other people from the Wasmuth lab, then our collaborators, Dr. Cameron Cotter, Molly Tilley, and Dr. Constance Finney, Dr. Aurelia Correa, and Dr. Hongrui Liu, and of course, my family and the funding agencies without whom none of this would be possible. All right, so since I'm going to talk about these really interesting group of animals, myxozoans today, I always like to take a step back and look at all extant animals. See, all extant animals can be classified into 5 groups. This includes multiple phyla of bilaterian animals. This includes us, and 4 phyla of non-bilaterian animals. These are your sponges, ctenophores, placozoans, and then cnidarians. That's the group of animals we'll be talking about today. And in a way, the image that we see on the screen is a good representative of phylum Cnidaria as we know it. And these are cnidarians we are more familiar with, the large jellyfish, the hydra, corals, sea fans, sea anemones, and so on. But today I want to talk about a side of this phylum which we may not be that familiar with. And so this actually presents a more complete picture of phylum Cnidaria because it includes a group of animals called myxozoans. So who are the myxozoans? Myxozoans they are an enigmatic group of approximately 2,600 species, all of which are intracellular parasites, and all of which have significantly simplified their morphology compared to the free-living cnidarians. So these 4 new images here, these are myxozoans, examples of myxozoan animals. They can be found in both freshwater and marine environments, and they cycle between multiple hosts. And now, while myxozoans have been identified in birds, amphibians, reptiles, and even land-dwelling mammals, they are arguably best known for causing significant disease in fish. And here they pose considerable ecological, economic, and conservation challenges. Now, because of how different they look, it took nearly 200 years since the discovery of the first myxozoan species to place them within phylum Cnidaria. So on the left here, you see a phylogenetic tree from a paper in 2015, where they place this class Myxozoa in red over here, well within Cnidaria, which is this blue group on this phylogenetic tree. And just because they are not as well known doesn't mean that they are a small group of animals. In fact, nearly 1 out of every 5 cnidarian species is actually a myxozoan parasite. And that can be seen in this pie chart on the right-hand side of this slide, which shows the distribution of the number of described species in phylum Cnidaria, which is dominated by anthozoans here, followed by hydrozoans. And then the third most species-rich group in this phylum is actually myxozoans, with around 2,600 species. And another very important thing to know about myxozoans as we proceed is that all described myxozoans so far have a very interesting life cycle with multiple hosts. So they alternate between two free-living infectious life stages and two parasitic stages, and they usually in an annelid and a vertebrate host. In a general myxozoan life cycle, sexual development happens in the worm host, and following this, a free-living actinospore stage is released into the water column. The short-lived actinospore penetrates the epidermis of the fish upon physical contact, and it injects infective sporoplasm into the host tissues. And then the sporoplasm moves to wherever it wants to attack and live. And then the next stage, which is formed, is the myxospore, which is released usually when the fish dies. And the myxospore then is ingested by a worm host, an oligochaete host, annelid host, and then the cycle continues. And so why here in Alberta are probably the most well-known myxozoan is the one which causes a disease which we may have heard of called Whirling disease. And here I want to talk about this phylum Myxozoa because within this phylum, there's this genus called Myxopolis, which is the most species-rich group in Myxozoa. There are approximately 850 species described so far, among which the most well-known member is the parasite Myxobolus cerebralis, which is shown in this image over here on the left of the screen. It is a causative agent of whirling disease in salmonid fish like trout, salmon, and whitefish. Now, what this parasite does is it attacks the cartilage of salmonid fish. It impairs the nervous system. It leads to skeletal deformities. The fish may show color changes due to nerve compression, so the tail may actually appear dark or even black. And a whirling swimming behavior may be observed as the parasite invades the cartilage and impairs the nervous system. And this disease can be quite severe depending on the age and the size of the salmonid host. Usually young fish are most vulnerable, with mortality rates reaching up to 90%. And like other myxozoan species, it also needs two hosts. So in addition to its fish host, it needs this oligochaete called tubifex or the sludge worm as its definitive host. And Whirling disease is basically, it was first detected in Canada, in Alberta, in Johnson Lake in Banff National Park in 2016. And since then, it has been declared in four major watersheds in central and southern Alberta. This includes the Bow River, North Saskatchewan River, Old Man River, and the Red Deer River. Studies on whirling disease in Canada, while they're limited, there was a very thorough study which was done in 2019 in the Crow's Nest River. And the images which I've taken here are from the technical report, Whirling Disease technical report from the government of Alberta, which show that the results from this study showed varying degrees of impacts in different areas of the Crow's Nest River. They found that while the upper reaches of the river show little evidence of the presence of the parasite, the story is quite different in the other parts of the river. Because in the mid reaches of the river shows signs of moderate severity with a large number of young fish showing signs of this disease. And the lower reaches of the Crow's Nest River show high severity of disease. So in the left-hand side here in this map, areas in red are where the parasites are confirmed, where in yellow, these are areas at risk for infection. So in the lower reaches of the Crow's Nest River, where there's a high severity, the majority of rainbow trout in this area tested positive for whirling disease. And fish surveys here in this region showed a large age gap in the population along with a low number of adult rainbow trout. And what is also surprising is that the amount of parasite in the water samples which was taken from this river ranked among the highest levels amongst the highest levels compared to rivers in Colorado in the US, where rainbow trout populations declined by over 90% in a 5 to 10-year-old period. So here on the right-hand side, you see the number of parasites in 1 liter of river water, and the number for the Gunnison River in Colorado is followed by Crow's Nest River, the Fraser River in Colorado, and so on. So these numbers were quite high. And further, the Crow's Nest River rainbow trout are also sympatric with the endangered West Slope cutthroat trout, which are also susceptible to Myxobolus cerebralis, raising concerns for their conservation. And the rainbow trout is the third most valuable recreational fish species in Alberta, a sport that generates more than $1.1 billion per year for the province. So overall, this highlights the severe economic and conservation and ecological concerns this parasite poses in the province. Here, I want to show this image, which is from when the Johnson Lake was drained way back to counter this threat of myxozoan parasites. And this image on the right is a photo which warns people about whirling disease, and we need to clean, drain, dry your boat. And this is the one which I took in the Upper Kananaskis Lake when I had been there earlier last year. And this is by far not the only troublesome myxozoan in Canada. There are other troublesome myxozoans as well. One of the more well-known ones one is Kudoa thricetus that causes some problem in BC right next to us. And Kudoa thricetus is this myxozoan which is shown on this right. This is a myxospore of Kudoa thricetus, and it has a very broad host range, and it affects the muscle of over 30 different marine fish from several families around the world. And in BC, this includes the sea trout, herring, salmon, mahi-mahi, and so on. Now, this is an issue because when it infects the infected fish don't show visible pathology. But once the fish is harvested, once the fish dies, these myxozoans release enzymes which degrade the flesh post-mortem. So if there's low level of infection, it can go unnoticed in commercially sold fish, but in higher levels of infection, this high proteolytic activity results in extensive liquefaction of the muscle tissue, and the fish meat is no longer commercially viable. And finally, before moving on to Myxobolus rasmusseni, I also want to touch upon a lesson to be learned from across the border. And this is when I was there, this story came out. That was in 2016, when the Montana Fish, Wildlife and Parks Department in the US had to close access to over 180 miles of the Yellowstone River. And activities like fishing, swimming, floating, everything was prohibited. It was kind of a drastic step because its fishery contributes to more than $150 million each year to the local economy. And this closure resulted in a loss of over $500,000. And this was necessary because these fish were being infected by a myxozoan called Tetracapsuloides bryosalmonae, shown here in these arrows in this image on the right-hand corner. And it caused something called as proliferative kidney disease. All right, so let's move on to the organism that we have been working with, which is Myxobolus rasmusseni, which is an emerging myxozoan parasite in Alberta. And here, I want to talk about this. It's the myxospore of this organism is shown on this left over here. And essentially, when Dr. Cameron Goter and his colleagues, I discovered this emerging myxozoan parasite in fathead minnows in Alberta. And here in the middle image, you see a fathead minnow with these conspicuous lesions caused by Myxobolus rasmusseni infections. And this is named Myxobolus rasmusseni. And in addition to fathead minnows, this parasite needs tubifex, the same sludge worm, which is required by Myxobolus cerebralis as its definitive hosts. So what this parasite does to the fathead minnows is that it causes lesions to form around the eyes and nose, and causes large swellings of the operculum and pectoral girdles. And these lesions aren't harmless because they cause impairments to vision, olfaction. They affect the overall performance and also affect the survival of these infected minnows. So it's clearly evident that myxozoans have this negative impact, but myxozoans remain extremely understudied. So of the 2,600 rough or described myxozoan species. Only 8 of them have a genome. Only 14 of them have a transcriptome available. And even when we look at the basic biology of these myxozoans, only 50 have partial life cycles determined, and only 5 which have been confirmed in the laboratory. There are no known drugs to treat myxozoan infections and no known vaccines to counter these diseases. And this is a big gap in knowledge because genome and transcriptome data, it plays an important role in finding strategies to stop parasitic infections, and also to understand how they interact with their hosts. Because as more and more myxozoan genomes and transcriptomes are available, this facilitates comparative analyses, and scientists can pinpoint genes that could present potential drug targets or vaccine candidates. And I want to give one example of how this can be very useful and why we need to sequence more genomes of these parasites. Well, based on the available genomes at the time and comparative analysis between them, researchers were able to identify a category of proteins in myxozoans which could be potential drug candidates, and these were proteases. And based on this, there was a recent study which showed that which targeted a particular protease, a serine protease in Myxobolus cerebralis, um, in the annelid host of this parasite, which stopped the Myxobolus cerebralis life cycle, showing that this protease played a very important role in its infection. And at the end of the paper, they write, and I quote, genomic, transcriptomic, and proteomic analysis would facilitate further target identification for silencing using RNAi-based therapeutics. So just one example of how having this genomic data can be very helpful. All right, so what were the goals of our analysis, of our project? Well, we want to develop genomic and transcriptomic resources for 2 myxozoan parasites in Alberta, that is Myxobolus cerebralis, the causative agent of Whirling disease, and Myxobolus rasmusseni, which was recently discovered. And then we want to utilize the developed resources to explore strategies for parasite control and to understand how these parasites interact with their fish host. And for that, we started with sequencing the genome of Myxobolus rasmusseni. So the lesions in fatted minnows, which contain myxospores, were provided by Dr. Cam Goter, and we extracted DNA from these myxospores. And this kind of took we need to try and test different techniques. And I want to acknowledge all the hard work of Dr. Aurelia and Dr. Hungry from Dr. Finney's lab for helping with this step. And we were able to extract DNA from the myxospores. And then we sequenced a genome using the Oxford Nanopore long read sequencing technology. For this, we used a MinION platform. And it's interesting because this is a very small piece of equipment. Here we see another different machine, very similar to the MK1B, in which we used to sequence the genome. So that's how incredibly small some of these sequencers have gotten. And we sequenced the genome. And at that point, a very big issue with myxozoan genomics is contamination. That is, since we get these myxozoan samples from the host, there'll be a lot of host tissue surrounding it. We need to make sure that we remove all possible contamination using other techniques. So what we got lucky here, because just before we started sequencing this genome, the draft genome of the fatted minnow was available. So we essentially took our sequenced reads and we mapped it to the fatted minnow genome, which was available. And all the reads which mapped to the fatted minnow genome were discarded as potential contamination. And we only kept those reads which did not map to the fathead minnow. And then with those reads, we assembled the genome using a software called Fly. We also tested different genome assemblers and picked the best one. Following this, we polished the genome using a tool called Medaka. And then we further removed any additional contamination, non-host contamination, using a tool called Block Tools. And after we had After we had this genome with the contamination removed, we identified and masked repetitive regions in the genome using repeat modeler and repeat masker, and then annotated the genome, that is, predicted protein coding sequences within the genome using a tool called Breaker. And now I want to share some interesting things which we found from the myxozoan Myxobolus rasmusseni genome, which we have sequenced, and what we learned from it. So we found that the myxobol at 185 megabases, the Myxobolus rasmusseni genome was the largest myxozoan genome sequenced so far. So on the left-hand side of the screen, you'll see a graph which has a number of contexts into which the genome was assembled on the y-axis and the genome size in megabases on the x-axis. The green dot here at the very right-hand corner, bottom corner, is the genome of Myxobolus rasmusseni. And here is the region where we want to be. A good assembly will typically be assembled into the least number of contexts and right over here. So this is where we want to be. And you can see that myxozoan genomes are quite diverse in quality. For instance, if you look at the genome of Enteromyxum leae, another myxozoan, its genome is about 60, 70 megabases large, but it's assembled into around 70,000 contexts. So the assembly is not as good as Myxobolus rasmusseni. There's only one more myxozoan genome, which is sequenced using the long read technologies, which is Myxobolus honguensis over here, which is similar in quality to Myxobolus rasmusseni. So we kind of see that having a long read assembly kind of improves the assembly of myxozoan genomes. And I'll talk more about it. In addition, we also identified the mitochondrial genome of Myxobolus rasmusseni. And this is kind of important because this represents, we can use the mitochondrial genome and the mitochondrial genes to identify and monitor this parasite in Alberta. But it's also interesting to know that we can find this mitochondrial genome because it's only the second mitochondrial genome from a genus Myxobolus, which has 850 species. Right? And it's very important to know that at least one myxozoan has completely lost its mitochondrial genome. And so it's nice to know that Myxobolus rasmusseni has a mitochondrial genome. It's a large mitochondrial genome. It has a reduced set of mitochondrial genes. And yes. And then when we we masked repeat regions using repeat model and repeat masker, we found that the Myxobolus rasmusseni genome is extremely repetitive. And here on the left-hand side, you see on the y-axis are species names. These are invertebrate animals. On the x-axis is the percentage of the genome with repetitive regions. And this is from a paper in 2023 where they sequenced the genome of the Antarctic krillfish and found that it was highly repetitive, around 70%. And we find that Myxobolus rasmusseni has nearly 69% of its genome comprised of repetitive regions. And it's important to, and what this kind of tells us is that we really need to use long read sequencing technologies for myxozoan genomics because these long read technologies are much better at assembling repetitive regions. And so a lot of the myxozoan genomes, which are very small, we could be underestimating their size because the short read technologies are not able to assemble those troublesome repetitive regions correctly. And further, it also furthers our understanding of myxozoan biology. What is this highly repetitive region doing? And it's kind of the first step in developing strategies to kind of understand and contain them. So one of the things, and this is an ongoing story, but one of the interesting things that we noticed in this is that there was an expansion of family of DNA transposon derived transcription factor called flywitch transcription factors. And these are factors which are kind of rare in animals. They are not they are found in animals, but they are rare in animals. And only in another group of non-bilaterian animals called ctenophores, they have undergone expansion. So on the right-hand side, you see a tree with the number of flywitch domains on this pink bubbles. And even with ctenophores have only just 16, and in Myxobolus rasmusni, we found 55 of these transcription factors. And so we need to study them further. After this, we identified protein coding genes within the Myxobolus rasmusni genome using Breaker. We identified over 10,000 protein coding genes. We also did this for other myxozoan species, genomes which are available, because many of them do not have uh, their annotations available, publicly available. So we developed resources to promote comparative analysis among myxozoan genomes available so far. So that's shown on the left-hand side. That's the number of genes, protein coding genes found in each myxozoan's genome over there using the same tool called Breaker. And on the right-hand side, we show results of BUSCO analysis, which is identifying which shows how complete a genome is. It looks at 255 genes which are found in most eukaryotic genomes and looks at how many of them are present in the genome of our interest, and how many. And so what we see here on the right-hand side is the BUSCO result for all these myxozoan genomes. And Myxobolus rasmusni and honguensis, the long-read assemblies, they are some of the most complete myxozoan genomes available so far. Again, showing the advantages of using long-read sequences for myxozoan genomics. So at the very last, I also want to talk about how we can give examples of how we can use these developed resources to potentially identify drug targets and to know more about how this parasite interacts with its host. So first, to even do this, I'll analyze phylogenetic relationships within myxozoans, and we wanted to see where Myxobolus rasmusni lies. For this, we looked at all the single copy orthologs between myxozoans and free-living cnidarians, which is on the left-hand side here of this screen. And we built this phylogenetic tree where all myxozoan species grouped together, and we found that Myxobolus rasmusni grouped together with Myxobolus honguensis here. And so with these relationships, we could now look for gene family evolution to identify interesting examples of gene family evolution, like gene family expansion. And these are particularly interesting because myxozoans are known to lose very well conserved eukaryotic genes. So gene loss isn't that interesting, but gene family expansions are particularly intriguing because that means that they are performing an important and potentially interesting role in this group. And in one such example of gene family expansion, I want to give, which is on this right-hand side, are these serine protease inhibitors in cnidarians. So these serine protease inhibitors, also called as serpins, they play an important role in parasitic organisms like defense against host proteases in Schistosoma mansoni, and also act as immunomodulators in certain parasitic nematodes. And in a recent study of Myxobolus cerebralis, they found that some serpins they represent good targets for inactivating the parasite at an early stage in the fish host. And what we find is that this family of serpins has undergone expansion in Myxobolus rasmusni and Myxobolus honguensis. So here we find orthologs of potential drug targets which have been identified in Myxobolus cerebralis. So that's interesting. But here we also find that in one particular serpin 1 from Myxobolus cerebralis has undergone duplication multiple times in Myxobolus rasmusni and honguensis. And so these need to be studied further. And also, finally, I wanted to give an example of how it can further our knowledge of myxozoan biology. On the left-hand side, we show a phylogenetic tree of all proteins which contain the hexokinase domains in free-living cnidarians and Myxobolus and myxozoans. And we find that both Myxobolus rasmusni and honguensis had more proteins containing these hexokinase domains than free-living cnidarians. And since hexokinases catalyze the first step of glycolysis, this kind of increase in hexokinase repertoire in the parasite could reflect its kind of an increased reliance of the parasite on glycolysis to compensate for a reduced and inefficient oxidative phosphorylation system. And that's because myxozoans also have lost many subunits of its oxidative phosphorylation system, which it uses to make energy in the mitochondria. So right-hand side, we show how many homologs of known human mitochondrial proteins were found in free-living or cnidarians on the top and myxozoans on the bottom. And we can see that the number of human mitochondrial proteins with homologs and myxozoans is much lower than that in free-living cnidarians. And so this could represent a potential future. This can be a lead for future anti-parasitic strategies, as hexokinase inhibitors have been proposed in other parasites as potential drug targets. All right, so this is an ongoing story, and there are several aspects that will be explored in more detail in the near future. But essentially what we want to do is, now that we know we have a pipeline to go from a myxospore from like DNA to the genome analysis, we will use the same methods and techniques we use for Myxobolus rasmusni to develop genomic resources for Myxobolus cerebralis. We'll sequence its genome, transcriptome, and then we'll compare the genomes of the 3 Myxobolus long-read genomes to first explore host parasite interactions, how these parasites might be interacting with their hosts, and also to identify strategies for myxozoan control. And here we want to use this this generated data to focus on this genus Myxobolus, which contains so many disease-causing myxozoans. And here is where the generated data will also make all of this data available in a more user-friendly way for scientists to carry further analysis of myxozoan biology. So thank you. And yeah, I'll be happy to answer questions.
Excellent. Thank you, Viraj, and thank you, James, for a really exciting and interesting presentation. We have come to the end of our time, so I just want to let people know that our next science seminar will be on February 13th, where we'll be hosting Dr. Edward Bork from the University of Alberta, who will be presenting on the role of grasslands in providing carbon storage, an important ecosystem service of carbon storage, as well as the potential beneficial role of livestock grazing in enhancing that carbon storage potential. So I hope you can join us on the 13th. If you are not getting regular updates for these seminars, please send an email to the Office of the Chief Scientist at aep.ocs, and that's at gov.ab.ca. And with that, I just want to thank again James and Viraj for sharing your work with us today. That was fascinating to see, and we look forward to exciting results in the near future as well. Thank you very much.